Hello, my name is Brianna Cummings. I'm a 12th grade student at Biotechnology High School, and the name of my project is The Microbiomes of Man's Best Friend, How Truly Different It Can Be. Aside from their human counterparts, people are known to have very close interactions with their furry friends. Some of these interactions, such as sharing food or even sleeping in the same areas, are considered unsanitary because animals are assumed to have mainly harmful bacteria. In this experiment, the plan is to prove that the variation in the bacteria found in the microbiomes of both humans and animals are actually pretty small because they live in the same environments. That being said, if this experiment was to happen in real life, animals such as cats that you might let roam the neighborhood or dogs that you might keep outside would not be good candidates for the experiment. Swabs will be collected from the external auditory canal and the oral cavity from both species. Keep in mind, only humans are being tested, and on one side at least, and only dogs and cats are being tested. So it wouldn't be other household pets such as birds, hamsters, etc. On to my methodology. The first step would be forming an IRB so I can get its approval for live subject testing. Second, I would have the owners collect mouth and ear samples of both their pets or the pets that would be willing to have this done and themselves. The third step would be growing cultures from the swabs in an incubator, of course. And fourth, it would be to test the similarities. Now, with testing the similarities, we would do a t-test on the bacteria. The plan, if this experiment was to actually happen, was we were going to use the 16SR RNA gene PCR as a tool for identification of the bacteria present, so we would be able to get a pretty good understanding on the similarities and differences and how they compare in both areas. The desired outcome from the test is that the auditory canal would have more similarities in the bacteria found because it can't be changed by the dietary restrictions for animals and humans. For example, of course, you can't feed your dog chocolate. You might have eaten chocolate earlier in the day, and that's going to change just the makeup of the bacteria found, because maybe you might find bacteria or more bacteria in the human's mouth that might be drawn to carbohydrates more than you might find in the animals, which would be drawn to the protein that the animal is eating from its food. There are three results that could have happened if this experiment was actually conducted. The first one was when the bacterial sequencing results are compared, the external auditory canal samples will have more similarities between species because the bacterial makeup of the region cannot be altered by aspects such as diet. Of course, what you eat can vary, but chances are what you put in your ears will not vary. The only thing that humans really do that might change it a little bit is when humans clean their ears, and even then, that might not alter the bacterial presence too much. Second is when the bacterial sequencing results are compared, the oral cavity samples will have more similarities because of the shared omnivorous diet between the two species that share the living space. Now, if this experiment actually was able to happen and I could expand upon my research, I would also compare the diets that the humans have compared to their animals, because not all humans are omnivores. Well, they can eat it, they can process it, but not all people choose that diet. Third, when the sequencing results are compared, the human oral or auditory canal will hold slightly more bacteria than their animal counterparts, but they will hold relatively the same amount of harmful bacteria. Now, harmful bacteria is still to be determined because I did not get to do all the development on this project that I wanted to with Dr. Schuster in class. But basically, number 3 is the one that I believe will actually happen if this was to be conducted, only because animals are held in the same environment, but humans are allowed to go out into the world and interact and contract more bacteria from outside and bring it in than animals are. Of course, animals go out occasionally, but if it's a house animal that might just go out in the backyard, it's not exposed to too much. The selected outcome, number 3, was chosen because the entire point of this research was to debunk the stigma that animals are dirty in comparison to humans, and it was trying to dive deeper down into the question of what is dirty on a microbacterial level. From this research conducted to build my experiment proposal, option number 3 seemed like the most realistic outcome that could have been chosen. This project and its results can not only encourage potential pet owners who are reluctant to have germy creatures in their residence to adopt their own furry friends, but hopefully it can put current pet owners at ease knowing their germs that their friends have cannot hurt them, and maybe it could strengthen their bond within the household. This project is specifically important to me because I have 2 dogs and I have 1 cat, and I have a mother who also feels like they are germ ridden. And if she was able to see this on a numbers level, because she is a numbers person, she needs the facts, maybe she would get along with the animals a little bit better and trust them a little bit more. And the only acknowledgments I really have for this project is for Dr. Schuster, because before this project, I did not even know how to begin to test for bacteria. I've always wanted to, but he kind of made this a fun project for me and showed me how I could expand upon it